The primary component is actin. However, microfilaments interact with and are modified by numerous other proteins, collectively known as actin-binding proteins, or ABPs. These proteins regulate assembly and disassembly. Proteins like profilin and thymosin beta-4 influence G-actin availability, while capping proteins, for example, capping protein and CAPG, control filament length by binding to the barbed end. Cross-linking. Proteins like alpha-actinin, fascin, and fimbriin link actin filaments together to form bundles or networks. Branching. The ARP2/3 complex can bind to the side of an existing filament and nucleate the formation of new branched filaments. Severing. Proteins like gelsoline can cut actin filaments into shorter pieces. Motor proteins. Myosin motor proteins interact with actin filaments to generate microfilaments, also known as actin filaments, are the thinnest protein fibers of the cytoskeleton found in the cytoplasm of eukaryotic cells. They are dynamic structures that play crucial roles in various cellular processes. Composition. Microfilaments are primarily composed of a globular protein called actin, or G-actin. These individual actin monomers polymerize to form long filamentous structures called filamentous actin, or F-actin. Each microfilament is made of 2 intertwined strands of F-actin wound in a spiral. They are approximately 6 to 8 nanometers in diameter, making them the narrowest of the 3 types of cytoskeletal filaments: microfilaments, intermediate filaments, and microtubules. Like microtubules, actin filaments are polar. They have a fast-growing end, known as the barbed end, and a slow-growing end called the pointed end. Actin monomers, or G-actin bound to ATP, are added more rapidly to the barbed end. ATP hydrolysis to ADP drives filament assembly and disassembly, a process called treadmilling. Actin filaments are assembled into 2 general types of higher-order structures. In bundles, actin filaments are cross-linked into closely packed parallel arrays. These can be polar arrays, where all barbed ends point in the same direction. Non-polar arrays have barbed ends that point in both directions. They are often associated with contractile proteins like non-muscular myosin and are involved in moving substances within the cell. Actin networks. In networks, actin filaments are loosely cross-linked in orthogonal or right-angled arrays, forming 3-dimensional meshworks that resemble semi-solid gels. These networks contribute to cell shape and provide mechanical support. A specialized type of microfilament structure, periodic actin rings, can be found in axons forming tetramers with spectrin to link neighboring actin